Welcome back friends, welcome back in another video tutorial from microbiology with Charnik. So in this video tutorial we will going to discuss about some important MCQs from the chapter construction and screening of the genomic and cDNA libraries. So we have covered each and every topic of the construction and screening of genomic and cDNA libraries. There is a playlist available in my channel named construction and screening of genomic and cDNA library. So now we have, although only one topic left, that is chromosome jumping. I will also try to upload this chromosome jumping by tomorrow. So now, as we have discussed about each and every topic except one, so now it is time to discuss about some MCQ from that topic, right? So let's start. So genomic library construction is concerned with what? Genomic library construction is important so that we can isolate gene and study them whenever we are requiring, right? So they are important for gene isolating and isolation. We are not bothered about production of antibiotic via genomic library, no. Regeneration, no. Protein production, no, because we are simply preserving the DNA so that we can use them in for further purposes, right? So gene isolation, it is the topic which is most preferable on the basis of that question. Okay, now let's move to the next topic, that is what is probe. So probe means what? Probe means is a short single stranded DNA molecule which is attached with a radioactive isotope, right? And now this DNA can be chemically synthesized or can be naturally synthesized, right? So here the D option will be more preferable than the anything that present here, right? So either purified or synthesized single stranded DNA tagged with any kind of dye is called probe. Okay, third important one, which of the following is not termed as hybridization? So I want to discuss with you that if two DNA coming from male and female of the same species are together annealed, they are not considered as a hybridization. Hybridization is only in case of whenever two DNA from different species are annealing, or let's say DNA and RNA are annealing, or DNA and complementary cDNA are annealing. Okay, so D option is the correct one. So now the fourth question is, which of the following does not affect the formation of the hybrid DNA? So we know that ionic strength and temperature is very important for working of the DNA polymerase, right? Because it is an enzyme. But an homologous DNA is also important, because if homologous DNA is present, then homologous DNA means what? Same DNA, right? So hybridization occurs between two complementary DNA, not between two same DNA, right? So presence of homologous DNA does not affect the formation of hybrid DNA, right? Because it will not participate in hybridization, but only pressure have the ability to affect the hybridization. Okay. Now the fifth question, that is radio labeling is generally brought about by addition of the radioactive phosphorus. Yes, obviously with the help of T4 polynucleotide kinase. So the statement is true. Which DNA usually is restricted to making a genomic DNA library? Obviously the genome, we are using restriction in nucleus to genome, and then each of the fragments are cloned, right? And that is how we are getting a recombinant clones or a set of recombinant clones, right? That what we are referring as genomic library. But we are not cloning here plasmid, not fudge, not operon, right? So now the seventh question, which is the function of the polynucleotide kinase? I have told you to add the radioactive phosphorus group at the 5' end. So addition of gamma phosphate, that can also be, you can preferred that any kind of phosphate addition at the 5' end are called, or can be done with the help of polynucleotide kinase. Okay. Now, why is probe labeled? So remember that you may found some confusion between the topic between the option A and C, but let me clear you, we are also using radioisotope, okay, that you cannot visible until or unless you are using a autoradiogram, okay, and without help of probe you cannot visible any kind of DNA, okay. So probe do not improve visibility, okay, but it improves the location identification, that means where your DNA present, okay. And remember that probe can generate visibility. If the generate visibility option is present here, then we can consider also it, right, but it not improve visibility, because visibility can be improved to someone who already have some extent of visibility, but probe totally lacks it, okay. So improve location identification is the most appropriate option in terms of the question, okay. Now the process by which a probe is used to screen a library, here similar thing you can face, that hybridization and colony hybridization both are quite right, but colony hybridization is more accurate, that is why C option is correct, right. And the first genomic DNA library were cloned in plasmid, okay. So that was the 10th most important MCQs from the chapter construction and screening of the genomic and cDNA library. So we have only one topic left, that was that is our chromosome jumping. We will try to upload this by tomorrow. So till then, bye bye friends.